The last structure we're going to be looking at is the quaternary protein structure. So quaternary protein structure is the way two or more protein chains associate to form a single 3D unit. They're held together by non-covalent bonds mostly. Some of them will have some disulfide bonds, and sometimes they'll have a non-amino acid portion, like what we saw with the conjugated proteins. So an example of that is hemoglobin. So hemoglobin has 4 polypeptide chains, which are all similar to myoglobin. So we'll look at the structure here in just a second. And they're held together by hydrophobic groups, so that non-covalent bonding is taking place. And in those 4 chains, we have 4 heme groups, and they have an iron atom that is essential to its function. So oxygen is able to bind to the iron, and then so each hemoglobin is able to carry 4 oxygen molecules. So hemoglobin is, well, let me just show you the structure real quick. So here's the structure. There's that iron there in the middle. And so the oxygen is able to bind to that iron. And hemoglobin is a cellular protein, so that just means it's found in cells, and it's carried throughout the body in red blood cells. So it's an oxygen carrier in the red blood cells. Okay, collagen is another example of a quaternary protein structure. Here, collagen has, well, first of all, collagen makes up about 30% of all proteins in mammals. It's fibrous, so remember that just means that it's insoluble and it's tough, and it's the major constituent of skin, tendons, bones, blood vessels, and other connective tissue. Collagen has 3 intertwined chains that are about 1,000 amino acids each. So you can kind of see in this picture, it has all 3 chains wrapped around one another to form this stiff rod like triple helix. And what holds them together, what binds them together, the 3 different chains, is hydrogen bonds. So that's what keeps them there. And in collagen, glycine is every 3rd amino acid, because it's the only amino acid that's able to fit in the center of the tightly wound tightly coiled helix. So it's very, very strong. Okay, so just a summary of all the structures that we've talked about. So primary structure, remember, was just the sequence of amino acids in the peptide chain. So there's an example there for amino acids in the chain. So there's that's all that's included in primary structure. Secondary structure, we looked at 2 different possibilities. We had an alpha helix and a beta sheet. And excuse me, both of those were held together by hydrogen bonding with the backbone groups in the amino acid. So the hydrogen that's bonded to the nitrogen, and does a hydrogen bonding with oxygen on the carbonyl. And then we looked at tertiary structure, and we know that tertiary structure is formed by the interactions of the side chains. So we looked at specifically, there's hydrogen bonding taking place. There are disulfide bonds. There are salt bridges. If you have ionic bonding, you're going to get a salt bridge. There was hydrophobic interactions. So all of those taking place to form the tertiary structure. And then last of all, we looked at just now the quaternary structure, where we have 2 or more proteins or peptides that are held together by non-covalent interactions. We also looked at fibrous proteins, which were insoluble and tough, and globular proteins, which are water-soluble. simple proteins that can contain only amino acids, and then conjugated proteins have something other than the amino acid. So like for hemoglobin, we saw that iron was also in there, so that would be considered a conjugated protein. With, kind of continuing with collagen here, in collagen, the proline amino acid are hydroxylated in a reaction that requires vitamin C. So that's something that is a part of collagen. It makes it strong. It makes it tough for the fibrous protein. But if you lack that, if you lack vitamin C, because you don't have access to fresh fruit, like for armies or navies or medieval people, they're very susceptible to scurvy. So scurvy is just weak collagen is formed because that vitamin C is not there to be able to hydroxylate the proline amino acid. And so when that happens, when the weak collagen is formed, then that leads to skin lesions and fragile blood vessels. So you get things like swelling, blackened bruises, swelling, bleeding gums. So they've figured out that vitamin C will prevent scurvy or even cure scurvy. But then we have something else, osteogenesis imperfecta, another disease, but this is actually caused by defective genes. So this leads to weak bone structure. So with collagen, incorrectly synthesized, then you get weak bone structure. So this requires genetic testing, and there's no cure to it. So it's quite a bit different. I mean, it's some of the same ideas there with, the incorrectly synthesized collagen, but there's no cure for that. Okay, so just how we saw proteins or amino acids, can come together to form these proteins by the peptide bonds. We can also hydrolyze the peptide bonds and form amino acids. So it's just the reverse of the protein formation. So here we have 4 amino acids, and when they are hydrolyzed, the bond breaks, the peptide bond breaks. And then, so you can see for that first one listed there, that's alanine. So when that peptide bond breaks, the nitrogen then gains 2 hydrogen, and it goes back to having a positive charge, and then we gain an oxygen on the carbon with a negative charge. So it goes back to looking just like it did before the peptide bond was formed. So hydrolyzing, again, it's just the reverse of the protein formation. So to be able to hydrolyze proteins, this is done with usually an acid. So if you use an acid with heat, it will hydrolyze the protein. Well, if you use a base, then the base could destroy some of the amino acids. So really, an acid is recommended. A protein can also be denatured. So denaturation is just a disruption of the protein's shape, and it does not affect the primary structure. So you can see at the bottom, we have this protein, folded and everything, but if I heat it, then it denatures the protein. So the structure stays intact, the primary structure stays intact, but we lose any secondary or tertiary or quaternary structures. Those go away. So there's several different ways that we can denature a protein. They're all listed right here. We can heat the the protein and it will denature. So an example of that is heating an egg. Okay, if you heat up an egg, it denatures it. Mechanical agitation, that's another egg example. Whenever you're using a mixer and you're mixing the egg whites, they go from a liquid into this fluffy foam with the bubbles, the air bubbles held in place. And then we can use detergents, so low concentration of detergents disrupt the hydrophobic side chains. And then organic compounds, the polar solvents, interfere with hydrogen bonding. The pH change will interfere with the ionic bonding that occurs. So will inorganic salts also disrupt the ionic bonding. So all these things are able to denature a protein. And most denaturation is irreversible. Some of them will. Some of them can go, back to their original structure, but most proteins, when they're denatured, cannot go back to their secondary, tertiary, or quaternary structure. Denaturation also changes physical, chemical, and biological properties. So a lot happens with the denaturation.